Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt to more scientific basis for classification. He used simple morphological character to classify plant into trees, shrubs, and herbs. He also divided into two groups, those which had red blood and those which do not. In Linnaeus' time, a two-kingdom system of classification with plantae and animal kingdom was developed that include all plants and animals respectively. This system did not distinguish between prokaryotes and eukaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals were easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time, though plants and animals have a constant under all different systems. The understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other organisms have also been understood differently by different scientific over the time. R.H. Whittaker proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetical relationship. Table 2.1 gives a comparative account of different characters of the five kingdoms. The three-domain system has been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the system in the detail in higher classes. Let us look at this five-kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification systems include bacteria, blue-green algae, fungi, mosses, fern, gymnosperm, and the angiosperm under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cell. This placed together groups which widely different in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, cyanobacteria, with the other groups which were eukaryotics. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas, Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic groups fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls while the green plants had cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae with plants and both having cell wall, with paramecium and amoeba, which were earlier placed in the animal kingdom which lacked cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but also its phylogenetic that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plants, animals, kingdoms respectively, will be dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in extreme habitats such as hot springs, desert, snow, and deep ocean where very few other life forms can survive. Many of them live in or know other organisms as parasites. Bacteria are grouped under four categories based on their shapes: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrium, and the spiral spirillum. Figure 2.1. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most expensive metabolic diversity. Some of the bacteria are autotrophic. They synthesize their own food from inorganic substance. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of the bacteria are heterotrophic. They are depend on the organism or on dead organic food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty, halophiles, hot springs, thermoacidophiles, and marshy areas methogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characteristic by the presence of a rigid cell wall and, if motile, a flagellum, the cyanobacteria. Also referred as a blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. Figure 2.2. The cyanobacteria are unicellular, colonial or filamentous freshwater marine or terrestrial algae. The colony are generally surrounded by a glytinous sheath. They often form bloom in polluted water bodies. Some of these organisms can fix an atmospheric nitrogen of specialized cell and called heterocyst. Example: nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers of many of them have a significant impact on human affairs. They are helpful in making curd, milk production, antibiotics, fixing nitrogen in legumes, roots, etc. Some are pathogen causing damage to human beings, crops, farms, animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to another. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista. All single-cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistian to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoan under Protista. Members of the Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the protistean cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually and sexually by a process involving cell fission and zygote formation. Chrysophytes. This group includes diatoms and golden algae tasmine. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, planktons. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping shells which fit together as in a soap box. The walls are embedded with a silica and thus the walls are indestructible. Thus diatoms have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of the years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producer in ocean. Dinoflagellates. These organisms are mostly marine and photosynthetic. They are appear yellow, brown, blue, or red depending on the main pigments present in their cell. The cell wall has a stiff cellulose plate on the other surface. Most of them have two flagella. One lies longitudinally, other transversely in a furrow between the wall plates. Very often, red dinoflagellates, example Gangulex, undergo such as rapid multiplication that they make the sea appear red tides. Toxin released by such large numbers may even kill other marine animals such as fishes. Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long one, though they are photosynthetic in the presence of sunlight. When deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, example Euglena. Slime molds. Slime molds are the saprophytic protist. The body moves along decaying twigs and leaves, engulfing organism material. Under suitable conditions, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms floating bodies bearing spores at the tip. The spores possess two walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, sea water, or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shells on their surface, some of them such as entamoeba and parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases and such as sleeping sickness, example trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of the thousands of cilia. They have a cavity gullet that opens the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be stirred into the gullet. Example Paramecium. Sporozoans. They include diverse organisms that have an infectious spores-like stage in their cell life cycle. The most notorious is Plasmodium malaria parasite which causes malaria, a disease which has staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They have shown a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make bread or beer. Other fungi cause disease in plants and animals. Wheat rust causing Puccinia is an important example. Some are source of antibiotic. Example penicillium. Fungi are cosmopolitan and occur in air, water, and soil and animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With exceptions of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long cylinder thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called syncytic hyphae. Others have septae or cross holes in their hyphae. The cell walls of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate, and others are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae lichens and with their roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores, media, or sparozospores or zoospores, and asexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following three steps. Fusion of protoplasm between two motile or non-motile gametes are called plasmogamy. Fusion of two nuclei called karyogamy. Meiosis in zygote resulting in haploid. Hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cell 2n. However, in other fungi, Ascomycetes and Basidomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reproduction division occurs, leading to formation of haploid spores. The morphology of the mycelium mode of spore formation and fruiting bodies forms the basis for the division of the kingdom into various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasite on plants. The mycelium is aseptate and syncytic. Asexual reproduction takes place by zoospores motile or by aplanospores non-motile. These spores are endogenously reproduced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar in isogamous or oogamous. Some common examples are Mycor, Rhizopus, the bread mold mentioned earlier, and Albuco, the parasitic fungi on mustard. Ascomycetes. Commonly known as sac fungi, the Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast, Saccharomyces. They are saprophytic, decomposer, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The sexual spores are conidia produced exogenously on the special mycelium called conidiospores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are Aspergillus, figure 2.5b, Claviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered as delicacies. Basidomycetes. Commonly known form of Basidomycetes are mushroom rabbit fungi or puffballs. They are grown in soils, on logs, and tree trunks, and living plants' bodies as parasites. Example: rust and smuts. The mycelium is branched and septate. The sexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about fission of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom, figure 2.5c, Astimacus mud, and Puccinia rust fungus. Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belongs to. It is also possible that the sexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi got correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of the member of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The Deuteromycetes reproduce their mycelium is septate and branched. Some members are saprophytic or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Coletrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae include eukaryotic chlorophyll-containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasitic bladderwort and Venus flytraps are examples of insectivorous plants, and Cuscusta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycles of plants has two distinct phases, the diploid saprophytic and the haploid gametophytic, that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomena is called alternation of generation. You will study further details of this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by a heterotrophic eukaryotic organisms that are multicellular and their cell-like cell wall. They directly or indirectly depends on plants or food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient features of various phyla are described in chapter 4. Viruses, viroids, prions, and lichens. In this 5-system classification of Whittaker, there is no mention of lichens and some cellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effect viruses can have on us. Even if we do not associate with a condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular structure organisms that are characteristic by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell as replicate themselves killing a host. What you call virus living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as casual organisms of the mosaic disease of tobacco, figure 2.6a. These were found to be smaller than bacteria because they pass through bacteria-proof filters. M.W. Bijenrik, 1890, demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as a contagium vivum fluidium, infectious living fluid. W.M. Stanley, 1935, showed that virus could be crystalline and crystalline consists largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, virus also contains genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses, figure 2.6b. The protein coat of cold capsid, made of a small subunit called capsomas, protect the nucleic acid. These capsomas are arranged in helical or polyhedral geometrical forms. Viruses cause disease like mumps, smallpox, herpes, influenza. AIDS in humans is also caused by a virus in plants. The symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunt growth. Viroids. In 1971, T.O. Dinner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroids. The RNA of the viroids was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size of viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant CR Jacob disease (CJD) in humans. Lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is also known as phycobiont and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if